As described in another video, HFR plasmids in bacteria can insert into the bacterial chromosome at a specific site, and that becomes important. And then from this site, when they attempt to replicate, they actually then begin to drag the host bacterial chromosome along with them, so that this will result in a high frequency of recombination, hence the name HFR, of the genes on the chromosome of the donor strain, which has the HFR plasmid, to the recipient strain, which does not. As I describe in another video, an experimental design often has to control for a number of scenarios. And so in the videos which follow, the donor of the HFR strain would need to then have some metabolite that it can't synthesize. And this metabolite, let's say biotin, for example, would not be present in any of the petri dishes where the strains would be inoculated, so that the HFR donor strain would not survive on any plate. One could also then assure that the recipient strain had not received an HFR strain, so they were not now HFR plus, and that the mutant genes maybe were deletion mutants, which could not revert to the wild type condition. These are just examples of things which would need to be controlled for the next videos. With that being said, here's a chance for you to map a bacterial chromosome. Let's imagine that the purple strain has an HFR plasmid, and that it is His plus, in which it can make its own histidine. It does not have to be supplied histidine. Lys plus, it can make its own lysine. Arginine plus, it can make its own arginine, and methionine plus, it can make its own methionine. So these metabolites do not have to be supplied in the media. Now, this purple strain is an auxotrophic strain. It is missing the ability to make biotin, and since none of the plates given here would have biotin, that purple strain would die. So even though it has histidine, lysine, arginine, and methionine synthesis genes, it would not be able to survive on the plates as given. The pink strain can make its own biotin, so it would be able to survive on all of the following plates. However, each of the plate is then missing an additional metabolite, such as histidine, lysine, arginine, or methionine, and the plates are indicated. So the HFR strain is mixed with the non-HFR strain. They are allowed to conjugate for varying amounts of time. So you will see that there are 4 intervals here: 10 minutes, 20 minutes, 30 minutes, 40 minutes, at which point then in the original experiments a blender was used, but obviously then you could use some sort of shaker bath, which would then end the conjugation, and then the bacteria could be plated out onto these 4 plates. The purple strain would not survive because it does not make biotin, which is missing in these plates, so it would fail. And the only bacteria of the pink strain which would survive are those which have undergone conjugation and received a gene for a protein to synthesize something that the plate is lacking. So only those bacteria which have received a new gene for histidine synthesis would be able to survive on the plate without histidine. Only the pink bacteria which have undergone conjugation and received a gene for arginine synthesis would be able to survive on a plate without arginine. Based on this, and keeping in mind that HFR plasmids insert into a specific site in the genome, how would you map the position of these 4 genes on the bacterial chromosome?